Can anyone go to Kennedy Elementary School tomorrow morning at 8 o'clock and give away some ducks?
Okay.
All right, great, thanks. We're giving some away earlier than what we normally do at Madison and Kennedy. I'm going to Madison, but I can't be at two places at one time. And anyway, so that'd be great. Thanks.
Wonderful. What great news. Thank you, Craig, for working so hard on getting that donation from the Norman Rotary School Foundation. That's wonderful. Jennifer Willis, Beckett, sorry. I wrote Willis. I'm so sorry. I wrote Willis a long time ago. I'm so sorry. Has an announcement.
I'm going to kind of turn this way. So if you're not too tired on Sunday, please come out to Farm Girl Fair. It's out at Sandy River Ranch. You take 72nd. It dead ends. No, Tecumseh. Out to 72nd. It dead ends in their driveway. So if you haven't heard of Farm Girl Fair, it's a really awesome kind of little. It's almost like a little county fair, but it's at Sandy River Ranch. I'll be there selling my jams, farmer daughter jams. Everything has to be homemade and nothing's mass produced. There will be about 60 different artists and vendors. They'll have a pie eating contest. They're going to have a kids vendor area. They'll have entertainment. They'll have food trucks. I have about 12 different jams, but I'm super excited this year. I've got a Save the Boobies jam. It's a caramel apple flavor, but all of the proceeds for that will go to breast cancer, specifically to enhance breast cancer patient care with Norman Regional.
And with no further ado, can I have Zane Woods come up here? The reason we did not have a guest today is because we're inducting a new member. Well, I should have this memorized by now. Okay. Mark couldn't join us today, but we already had his packet, and Zane was excited, and so he said it was okay to move forward with it. So, on behalf of the members of the Norman Rotary Club, I'm delighted to extend a warm welcome to our newest member, Zane Woods. His sponsor was Mark Crittenbury. Joining Rotary is not just about becoming a member of a club. It's about becoming part of a global community dedicated to making a positive difference in the world. As a Rotary Club, we uphold the values of service, integrity, diversity, and leadership. We are united in our commitment to improving lives locally and globally through our various service projects and initiatives. Zane, we are excited to have you join our club on this journey through service and fellowship. Your unique skills, experiences, and perspectives will enrich our club and contribute to our collective efforts to create a lasting change. As you embark on your Rotary journey, you will have the opportunity to connect with fellow members, participate in meaningful projects, like Rotary Road Rally Festival maybe. That's right. And engage in activities that will align with your passions and interests. I'm not sure about the league in bodybuilding, but you can teach us more about that. We encourage you to take full advantage of all that Rotary has to offer and share your ideas and aspirations with us. Once again, Zane, welcome to the Norman Rotary Club. We are thrilled to have you as part of our Rotary family and look forward to working with you to make a positive impact in our community and around the world. Okay, wonderful, thank you. Very excited, y'all. Please make sure y'all welcome him at the end of the meeting. And Stephen Correnda, are you introducing? Yes. All right. No further ado, I'll bring Stephen Correnda up to introduce our speaker today.
So how many of my fellow Rotarians are aging? You're getting older? Anybody? Okay, a couple of you. I love it. How many are concerned about your health as you get older? Good, we got the excellent, perfect speaker for you today. It is my delight to introduce to you Vivek Bajpai. He is with the School of Sustainable Chemical, Biological, and Materials Engineering. I think that's the longest longest named school at OU, isn't it? Yeah, okay. He's an assistant professor at that school. He has a bioengineering laboratory, and his laboratory uses state-of-the-art technologies of genetic engineering, such as CRISPR. You're going to have to tell us what that is. Okay, all right. This engineer sells DNA for various applications related to human health. I'm way beyond my biology class in my sophomore year here. He has got 3 degrees, check this out. MLB Medical College in India, Indian Institute of Technology in Kanpur, and a doctorate from the University of Buffalo, the State University of New York. His research work has been published in the top journals in the fields such as Science, Nature Biomedical Engineering, Nature Genetics, Stem Cells, Cardiovascular Research, etc. And his contributions in the field have been recognized by several awards, such as Horizon Award from the U.S. Department of Defense and the Outstanding Investigator Award from the National Institute of Health. Wow. It is a privilege to have you, Vivek. Thank you.
Hello, yeah.
If you want to move around, you can.
Okay.
Take it down now.
I would prefer to stay.
Okay.
So thank you very much for this opportunity to speak about some of the work we do. And first of all, I would like to thank all of you here to be here. And as Liz was speaking about the core values of Rotary Club, one thing was standing out diversity, integrity, leadership, fellowship, which is really things that will be also uphold as we do our science or research or teaching experience. What we do in the lab, we call it basically cell engineering. Okay? And though this topic has been discussed in the context of human aging, but I would like to describe what we mean by cells so we can understand things in larger context a bit. And then I would also introduce some of the concepts which have been recently revolutionizing the medicine and different way we treat human diseases. So, first thing, this is the basic outline of my talk, and I will start with cells. Okay. So, what is a cell? Right. So, our bodies are made of cells. Okay. All of us, anything which is living being around us is made of cells. It is plant, our cat, dog, us. They're all made of cells. And if you look all across the life forms which you see in the planet, which is made of cells, right? But some of these cells, which we are made of, we are a collection of cells, right? So we can think of our bodies are made of cells. I don't know if I can show you some of these here. So the cells, but okay. So the cells, they basically make a small structure which is called tissues. We combine, cell combine to make tissues. Tissues makes organs, right? And organs make organisms, like whole of us. So we are a collection of cells ordered as tissues, and there's as we grow old, we also have the property changes in the cells, right? So our organs, they don't function that well, right? We see on top of it, some of these cells which are present in our body, these are, they don't actually repair much. For example, our heart tissues, right? Heart tissues, once heart is made, they have a limited repair capacity. So does the brain cells or brain tissues. But some of our tissues actually grow very well. So look at our hairs, right? When we are still young, our hairs, they keep growing, right? Our nails. So there are some cells in the bodies which actually keep growing, and our gut lining, it also has cells. They also keep us healthy because they keep renewing themselves. So these are called stem cells. Okay, so there are some stem cells present in the body which actually keep us more regenerative form. So I talked about cells, but what actually controls the cell function? What the cells do, right? So if you think, each of the cell has a nucleus in the center. That is called, this is called nucleus. So the part which I showed you here, this is a central, this is how a cell look like. And you have central nucleus, and then rest of the things like mitochondria, which produces energy. We need energy to run our bodies, and bunch of other things which are important for function. So the nucleus is the main, you can say it's kind of master of everything. So it does have something called DNA, which is called deoxyribonucleic acid. So this is a chemical molecule, okay? And this chemical molecule is present in all the cells. And this DNA molecule is actually very long. If you think it's about 1.6 meters long, maybe longer than a little bit. So, and our cells are very tiny. Our cells are, if I tell you that if you take a pen and put a dot on a paper, and if you break that dot into 1,000 small pieces, that is like 1 micron long. And our cells are about 10 micron long. And the nucleus is about 4 micron in diameter. So, this 1.6 meter long DNA, this is squared multiple times in order to pack and put it inside this nucleus. But what is important, a part of DNA is called genes. These are the segments of the DNA which is labeled as gene 1, gene 2. These are the genes which makes protein. So, our DNA, if you imagine DNA is $1, means 100 cents. The genes are only 2 cents of that whole $1 basically. So very small part of DNA make genes. But the important part is that genes actually carry message to make proteins. So genes make this messenger molecule called RNA, then we make amino acid, and then we get proteins. So each cell makes a set of protein at any given time. So as we have health or age or normal or disease, we have set of proteins. If the set of protein is good, that means we are healthy. If some of the proteins are bad, as we age, some of these proteins don't make very well, so we lose some of these proteins, right? So how can we make sure that we keep making good amount of proteins and good proteins? That means we have to learn how to manipulate DNA, okay? How to change some of the DNA, because, and we do it all the time. If you think about, if I have a change in the DNA, then it will make a bad protein. And that protein is going to affect, us. Same thing when you might know that all of us, when we get birth, we get tested for several diseases. They check a newborn baby's blood to test like about 56 diseases which affect DNA. Okay, so if there's disease, we get some of those could be corrected. So this is all already in place for like many years, right? So, so to understand DNA, in order to fix DNA problems, you first understand DNA. So there were once, before I go there, I would ask you a question. This is a heart and this is a brain, okay? When we are born, we have same DNA in all our cells because we got these DNA from our parents, right? And we get 2 copies of DNA, one from mother, one from father. So all of our body cells have the same DNA. But the same DNA is making tissue, heart and brain in a separate places, right? How it is doing that? Because there are 20,000 genes in the DNA, and in brain cells there are different set of genes make protein, and in the heart cell there are different set of genes which make protein. So which genes are making protein in a given cell, that will decide whether a cell is heart cell, brain cell, eye cell, or healthy cell versus disease cell, right? So it's important to understand or appreciate this fact that DNA is actually controlled within, so it controls which protein is going to be made in different cell types, which is, I find fascinating. And you can attribute to this God or science, but I always find these kind of mysteries fascinating. So there was one thing I want to bring, that diversity is because of DNA. Okay, we all have some differences. So how much we are similar? If I look my DNA with this Stephen's DNA, we are 99.9% same DNA. Right? So 0.01% of the DNA difference is making me, and Stephen is Stephen, right? If I look everybody's DNA, we have only few places where we are different in the DNA structure, right? But that is enough to make us a diverse people, right? And same diversity you see all across different forms we see, right? So whether it's like species of parrots or dogs, you will find diversity all across. So this is just a point. So when people started understanding DNA, early 90s, people thought, okay, if we want to correct things in the DNA, we don't need to know what DNA is. So there is one good scientist at OU, his name was Bruce Rowe. So he actually was one of the pioneers in the field, along with many other labs across the US and the world. They tried to understand or sequence what actually DNA looks like and what DNA is. So based on his research and many others' research, people figured it out in early 2000 what are the DNA structure is. Okay, and that gave us many more people to opportunity to then correct that DNA. So they figured it out what the normal DNA sequence looks like, and he was one of the early pioneers in that area. So I will talk about two ways how we use DNA information and some of the technologies. We call it DNA technology, recombinant DNA technology, or CRISPR, to correct some of the DNA or use that information of DNA to make new drugs or medicines which we typically use. We actually use in some of them as we treat cancers or a bunch of different diseases. Okay, so let me see. I have some time. Okay, so something is called recombinant DNA technology. So what is recombinant DNA technology? Here I'm showing you a bacteria. This is a bacteria. It's a bacterial cell. Okay, so bacteria grows very fast. If you put your food outside, it will spoil in a few hours, right? If you don't freeze it so bacteria, microbes are present everywhere and they grow really fast. So some of early scientists have thought, what if we use bacteria to make proteins which can be used as drugs to repair things, right? So this is what was done very early, and then we keep using this approach where we take human gene. So let's say you have these red fragments here. You take human genes, right? And then you break those gene parts from the human DNA and put into the bacteria, and use bacteria as a factory to synthesize a protein which is humans are going to use. And actually, many of us who have diabetes, for example, they still do that. We use, I will come back to the slide, but this is a typical example where we take gene which comes from so diabetes, for example, right? You might know that people who have diabetes, they have to take some time with insulin shots, right? And they take it at any time when they eat food, pretty much, right? So insulin is basically produced in the pancreas, which is just behind our liver, right? And that insulin is the cells which produce insulin. Sometimes they don't grow very well in some of us because of some things. And when we lose insulin making capacity, then we develop diabetes. So when we eat food, that glucose level which goes up has to come down, right? So insulin actually controls that and does that. So if we don't take insulin, it's not good. So we take insulin shot. Before recombinant DNA technology, we used to get it from pigs or humans, sometimes cadavers, was their pancreas was taken after, and then we isolate purified insulin from them, and that was being used. Now, what we do now, we just took human pancreas insulin gene from human cell and put in bacteria, and then this is our bacteria which we put. I have some issues here. Okay, this is the cell which we took this gene which is red in color, blue things, put into the bacteria. Now bacteria has this blue part which is insulin gene. Now we grow this bacteria in a kind of fermenter, just like you make a flask and you shake the bacteria, you grow them like 2 hours, and you have tons of bacteria, like billions of bacteria. Or each bacteria is producing now protein which is insulin. Then we purify that protein from bacteria and we label in these vials and we sell them as an insulin drug, and we all use it. Okay? So this is coming from bacteria. Now, why we are using bacteria? Because bacteria are easy to grow, and then you can grow them fast, right? So if I grow these things in different organisms, it will take time. But this is one example how the information which is in DNA is helping us to treat disease. So now when we think of disease or treatment, we think a medicine as a pill, Tylenol for example. We think, what is medicine? Everybody thinks it's a pill. more and more than 50% of our drugs nowadays, they are not just pills, they are basically proteins, and we call them biologics, okay? Most of them, people who have cancer, sometimes they get treatment, they also take these proteins actually, because proteins, as I tell you, that everything in the cell is done through protein. So if you can make protein, or you change the DNA to make the right kind of protein, then you are healing, or you are creating therapies to heal. Okay, so other thing I will talk about CRISPR. So the CRISPR it's a long name, I sometimes also forget, but I will tell you this is technology. What it does, you can think of CRISPR as a scissor and a torso. What it does, we can change sequence of DNA. If somebody has a mutation, which led him some disease, like sickle cell anemia is one example, but there's several examples where newborns might have a disease just because they have a change in the DNA, right? So using CRISPR, you can fix that problem, you know. And one example I will give you is cancer, which is leukemia, blood cancer, right? So there is a concept of stem cell bank, where newborn blood, umbilical cord blood is collected and is stored. And we store it with a hope, not a hope, but if unluckily or unfortunately the person newborn in future develops a blood cancer, his stem cells which were there at the beginning of the birth, they still might not have that change in the DNA. So we can use those stem cells, or we can change the DNA in the stem cells, correct them, and put those stem cells in the bone marrow. That's where the blood is getting formed. So we can correct. This is called bone marrow transplantation. It's a very common technique has been using from 1960s, 70s. So CRISPR allows you to correct the changes in the DNA to again heal people if needed. Okay. So I will skip this slide, but I will tell you the application of all this technology where you can correct using these technologies different diseases. What the applications we have? We have different applications in different domains, right? So medicine is one we already know, right? Many crops actually, the plants or produce we collect, right? Many of them are actually genetically modified. They are called GMO crops, and they have been modified in order to either improve their quality. Sometimes you might have seedless watermelon, for example, right? There's no seeds in there. So all these things have been done, and this is done before like even I was born. So people have been doing this for a long time. We know that in like improved livestock, like people practice this. If you have livestock, people try to improve the breed by crossing them with a better breed. The same thing actually, similar things been tried using these technologies. Plant crops, making the crops which can resist drought or infection, all these things have been, scientists have done that. Other thing is disease correction, I briefly talked about it. So if someone has, let's say somebody developed cancer, you take his blood cells out and you correct that problem in the DNA. Put back these cells in, so you take the cancer cells out, you fix whatever the problem was, you grow them more outside and put back into the patient. And that's how you can treat them actually. And this practice is like widespread. Even we have team in scientists and doctors who you just see they perform some of those things. Now vaccine, many times people take vaccine, and vaccine development is also being encouraged by these technologies. Like COVID-19, during that time, scientists could produce vaccine within a year because of this technology, and they could provide people to use this. Diagnostic tests, for example, is another aspect where they test, actually our nasals take the swab from the throat or nose, and they test it. This is also largely developed by the same technologies which we just briefly touched upon. So one example I will give you here is pigmentation disease. As we age, we know we also have a problem in complexion sometimes, right? There are several diseases which affect us. So, and it's a big actually industry. If you see, it's like about $8.6 billion annual medicine people take to correct the pigmentation disease. So some of those using this CRISPR technology, we actually found which are the part of the DNA which are creating problems to make pigmentation, and we can try to control that or correct that if for example using those DNA changes we could correct or we can bring the complexion normally. So now we are trying to develop some drugs which could actually affect the DNA, and then we can bring the normal pigmentation based on people's skin tone and color, right? So someone is a lighter tone or less lighter tone, they could be corrected with that. So other thing is cardiovascular aging, right? So one last few more minutes. So cardiovascular aging is the one thing where as we age, our cells of the body grow older. So we are trying to now find what are the DNA changes happening, and that is making our heart cells grow like slow or having problems. So we can then correct those. I will skip this part. I will just bring this point OU Bioprocessing Facility. So this is a new facility launched in Oklahoma City by OU. And then what we do here is we develop these biologics, these proteins which could help cure cancer and other diseases. And there's a big team of people involved here. This is just like a facility tour. I just want to show you the pictures to know that this is state-of-the-art facility. Only few places in the world I would say we have, and then OU and the Norman, Oklahoma City is one of the places which we have been developing this. So this is a summary I briefly just discussed. That message is that DNA makes the proteins, and the proteins do all the work. The changes in the DNA could be fixed by these technologies, and they have a lot of application in drug development, biologics, and other things. And I am happy to take questions.
With the COVID vaccine, as quickly as we bring out a new vaccine, so how do you attach that new vaccine, or how do you attach the components to that protein?
So what people do typically just they take the virus from somebody who is infected already. They take the virus and then they take the DNA from the virus, and then they use that virus protein to sensitize, let's say, an animal or the cells. These cells will start recognizing that protein and start making a protein against that viral protein. We call it antibodies. And we could do it quickly because the CRISPR and these technologies allow you to take part easily, manipulate them, and that's what we could do it. Otherwise, the first vaccine was developed like it took like many years to build. So so we saw in a COVID time that that's what we could do.
The CRISPR thing, that's absolutely amazing. You can correct those DNA, those gene problems. Have you gone back even further how to prevent those problems from happening in the DNA? Are there things that we can do practically for that?
So yeah, I mean, we have not, this is a very great point, we have not looked individuals' DNA beforehand to see, because we do not have all that information, but there's something called 23andMe, many times people sequence their DNA to find ancestries and all that, and sometimes you might have access to that data and you can take a look. So people have found out actually that some parts of the DNA we can use computers to look and then see whether there's some parts are like having some likelihood, they have DNA sequence which is kind of bad or more likely to produce a cancer. So there are already some work has been already there for some of us, but because we all are unique in our way, right? So we have to look at everybody's DNA to find what uniquely somebody has or not.
So earlier in your presentation, you talked about taking stem cells from the umbilical cords
Yes.
and using those to fix diseases.
Right.
But later you talked about taking DNA from you as an adult, I guess, and using that. So what's the difference between?
So, okay, I skipped one part intentionally. One is something called like IPS cells, or called induced pluripotent stem cells. So recently, people have developed a technology where I can take, or people can take anybody's cells and then turn them like a stem cells. Okay, and if you can do that, then you can do a lot more things. So these stem cells are called pluripotent stem cells. They can make any tissue of the body. So let's say somebody has spinal cord injury after a roadside accident, and then spinal cord, brain, neurons and nerve fibers are damaged. So you can take person's cell, make into pluripotent stem cells, and then turn those cells into those neurons and put back into the patient. So this is called tissue engineering or regenerative medicine. I skipped that part, but yes, you brought this thing. There are like different ways of doing it, and this is very revolutionary. The person who did it was a Japanese scientist in 2006. His lab showed they got Nobel Prize in 2012, and it's a very remarkable technology because it avoids this stem cell ethical concern. Because in the past, there's embryonic stem cells which comes from actually human embryos, and they were ethically prohibited to use. So this technology actually bypasses, and you don't actually use embryonic stem cells. You take patient cells, turn them like a stem cells, and then use them to heal them. So which is cool.
One more question?
Thank you. You were talking about protein then as it down the line from the DNA and amino acids. What connection does protein at that point have with the kinds of, we had chicken for lunch? That is protein.
Yes. So DNA, which was like in the chicken cell, that allowed chicken to make that protein, and then we took mostly like muscles, most of the time muscles which have meat we call it, right? So that muscle cells are specialized cells. If I show you in my early point here, let me show you, let's go back if I can. So these are muscle cells basically, but the DNA in those cells were the same. But those cells knew through this, we call it gene regulation, which I haven't talked about. Through that, you can actually, I go my first slide here. So if you look, this one here is your muscle cell. These are the muscle tissues here shown. Cardiac muscle, heart muscle. So these tissues have a very special characteristic. They are important for contraction, movement. So the only connection between DNA from the chicken muscle cell, so it basically muscle proteins, which make muscles in those cells. That was the question I was saying, what is the difference between heart cells and brain cells? Because in brain cells, we have same DNA, it's going to make brain-like proteins, and the heart is going to make heart-like proteins. And that's where I find it fascinating, because science is not able to solve these mysteries yet completely, how it happens, and yeah, but that's not the question.
Thank you very much. That was very interesting and learned a lot today. So thank you so much for coming. We appreciate having you and learning about that. I also would like everybody. Yes, sir.
Make a comment about this?
Sure.
I suspect, I'm pretty sure, I'm the only person in this room that has a degree from that department.
Wow.
And,
That's such a long name.
and I got my degree by being able to draw by hand the graphical solution of how many theoretical distillation trays it would take to turn petroleum into gasoline.
That was also very interesting. Learned something about your club number today, and ask them about it later. That was very great. Thank you for adding that. Please all make sure you welcome Zane. I'd also like to thank everybody that did trash pickup last weekend. I was not in town, and neither was Liz McCallum. I couldn't come up with your last name. I'm so sorry, Liz. Liz too couldn't be there either. So we weren't there to represent, but looked like y'all had a great crowd and a wonderful time. If you can stay a few minutes after to help us stuff some bags for this weekend, it'll only take like 20 minutes and be really fun, and you might learn something new about a Rotarian. Also, I'm offering a $25 gift card for to a restaurant here in Norman, local restaurant, who sells the most tickets for this weekend. So, challenges out there. If you've committed and something comes up, let us know, because we're counting on you to be there. We look forward to everybody on Friday or Saturday, and you were adjourned.
I can't stay, I gotta go.